So welcome to this SIMBA final meeting. Both we are warmly welcomed to this here in person, here in Copenhagen, in a very nice place, and also those who are online. So we will have, today we will have, I will first give a short introduction to what the SIMBA were, what kind of objectives we had, and then we will go to the panel discussions about microbes and plants, and then microbes, food and human health, and finally the marine microbes. But what was the SIMBA then? So when we started the project, our motivation was the One Health, so to raise awareness of the potential microbes from different sources to transport food, our food system, and improve overall knowledge of the microbes from land and sea towards the market needs, and also to improve human and also human and animal health with nutritionally fermented products, and improve the overall sustainability, including climate change, mitigation and innovation capacity of the food system, and also bring new cost-effective commercial application to the market that assist different stages and processes throughout the food chain. So our aim was to take the global approach to develop concrete microbiome products that include strains, mixtures, food, feed, services, and also the processes and methods which are highly commercially potential, which will then benefit society through improving food and nutrition security and resilience in the food production. So we had 7 different work packages which included the collection of the different microbes, and to see how they can be used in soil and plant, in marine part, and also foods, and also in human health, and finally how they then be upscaled, and also what is the sustainability impact of those microbes. So we have totally now at the end, when we came to end, we had 21 partners. We have 23 when we started, and we have both universities, research institutes, and also a lot of SMEs in our consortium. And here are shortly our outcomes, what we have found so far, or what we have done and collected the information about. We have collected a lot of microbe from partners, and now we have this availability, and now they are available for stakeholders. We have formulated different microbial consortia to plant growth, and also develop, and also so that they can be used in agricultural part to reduce the fertilizations and chemicals in the agriculture. So in marine part, we have found marine microbes for food and feed production and processing, and also improved overall knowledge of the coastal microbial part and also salt marsh microbiome to promote saline agriculture. In food part, we have found new microbiomes towards new feeds and foods, and also tools to enhance the utilization of microbiomes in the plant production and processing. And also in human part, we have found that the changes in dietary changes indicate low risk of type 2 diabetes, and also some cancers, and also indication of the diet-derived polyphenols, so that they modulate the gut immune response. so that it is potentially to design the gut health foods. And then we have, since this was innovation action, we have also upscaled the processes to produce microbiome biomass, microalgae production, and fermented food. Tools to identify and preserve microbes, online user-friendly metagenomic platform for stakeholders, and also professional marketing for launching innovation microbiome production to the market. And also we have assessed the drivers and barriers of the microbiome application, both for farmers and consumers, and also analyze the sustainability of these applications. And it seems that the high costs is the most barriers to use these new products. And we have so far got 15 innovations for industry, farmers, and sciences. So they included these microbes to support and protect plant from disease, and through support to plant growth, as well as the delivery methods. So also other innovations like that, how the microbes can be used to microalgae in fermented for plant products, and also strategies to uptake the microbial innovations in food production, and also the pipeline for plasmid analysis from metagenomic. And we have had a lot of interaction and dissemination activities. Here are also some of them. So we have had a lot of different open seminars, lunch seminars with other projects, training, 3 different training courses. We have had national events for citizens, and also interviews in TVs, radio, and newspapers, and also participation of science policy forums in both national and EU level, and also a lot of interviews, keynote, and other presentations. So here are some pictures what we have, how we have met during our 5-year project. Well, thank you. So now there will be this first session, which is about microbes and plant. So we will have first Alex from University of Bielefeld, and then Søren will continue with the same part.
Thank you very much. So I will talk a little bit about work package 1, which is basically the bioinformatics work package of the project. So we will address the data which is available in the repositories, because one of the goals of the project was actually to not only process data which is being generated in the project, but also try to include data which is already out there. So for those of you who are not very familiar with sequence data, I put up this slide, which everybody else has probably already seen. But I just want to make the point that, like 20 years ago, if you wanted to sequence, it took like a whole day to sequence like 5 kilobases. 5 kilobases is like 5 microbial genes. And there was a rapid improvement in the different technologies. And nowadays, if you use a short read sequencing machine, you can generate like 16 terabytes of data in a single run. And this is only short reads. And there are also these long read technologies available now, which can generate the same amount of data in a single run. Of course, that has consequences in the amount of data which is available in the public data sets. So this graph is showing the publicly available data in the European Nucleotide Archive, in the ENA. And of course, it's growing exponentially, which you could basically see in the slide before. And we almost have like 4 petabytes of data available, and this is only looking at the metagenomic data. So this doesn't include any human data or anything else. The human data is probably like 3 times as much, which is also available. So this is only looking at metagenomic data. And what you also see is where the data comes from, the continent where the data was generated. And it's actually interesting when you see when you look at the bottom, the orange color, Asia is taking up or is improving or putting out more and more data compared to all the other continents right now. So how do you deal with 4 petabytes of data? Okay, this is actually another different view on it. So on the right side, you see the European data, about a quarter of the data, European data, and it's actually pretty much distributed across all the countries across Europe. And we tried to analyze data from European data sets, and we will show you 2 different examples later in the talk. Okay, so the objectives of the work package were to build on this pre-existing knowledge, the existing data sets, the existing sequences, and then use data sets which are related to like soil data sets, marine data sets, gut data sets, and so on, and see if we can actually harvest these data sets for the project. And we also wanted to make it available to everybody, right? So first we had to process it. So we built a workflow for processing it. I will show you the workflow in a minute. And then the idea is to make it also available. So the data, the results we have will be either deposited to the ENA again, which is actually not that easy, because it's not that easy to deposit data to the ENA if the data is not owned by you. So even if you analyze the data, right? If you take public data, you analyze the data, it's really hard to deposit the analysis results back to the public repositories. Okay, so we wanted to collate the relevant data set and process it. We wanted to make it available through an open access database, and we wanted to make analysis tools, analysis workflows available. And so this is the analysis workflow that has been developed during the project. It's pretty complex. I will not go into detail, just showing the different steps. It starts with the raw data at the very top, and then it has several steps of like quality control. There's an assembly step where you try to reconstruct the genomes, the microbial genomes in the data set. And then once you have a genome available, you can look at function, right? And this is a nice thing about the whole genome shotgun approach. You get the complete genome out of it. You can look for function. You can look for pathways and so on. So we made this data set scalable because there's so much data available. An important point was to make it scalable. And the way we do is actually we use cloud computing resources. So the whole pipeline works in cloud computing environments, because in cloud environments, it's easy to scale it up, right? You just start more virtual machines. You have to distribute your jobs on the different virtual machines. And this is actually what we focused on when we built this pipeline. And I think it's working pretty well. So we can scale it to like hundreds of virtual machines to process the data now. And then on the bottom right, you can't really see the web interface. So once the data is processed, we make it available through a web interface. And in the web interface, you can search for your favorite taxon, you can search for your favorite function, you can search for your favorite pathway, and see if these are actually represented in the data sets or not. Okay, so we have 2 different examples we wanted to show you today. The first example is a data set where we try to look at European soil and see what kind of microbes are actually present in the European soil. And it turned out, we were very enthusiastic at the beginning, right? Because there are so many data sets. We said, well, we can process like hundreds of data sets and look at all the soil types and see what kind of microbes are present. But when you start looking into the details, you notice that the metadata is actually missing for many of these data sets, which makes it extremely difficult to interpret the results. Of course, you can analyze the data and you can look at genomes, but for most of the data sets, like the soil type is missing, temperature is missing, whatever you can think of metadata is basically missing in the database, so you can't make use of most of the data sets. So at least at the end, we identified, I don't know the exact number, maybe 50 data sets from 9 different countries across Europe, where some metadata was available. And then we processed the data and looked at, in this case, the taxonomic distribution. This is what you see on the right. This is a taxonomic distribution of these different soil types, and you can already see some clusters, and the clusters are actually representing countries here, right? So countries or soil types. But I will not go into detail very much. And then we also looked at the MAGs, the metagenome assembled genomes, which we reconstructed from the data, and it turned out that these Taum archaeota were very abundant in some of the data sets, right? So we had a deeper look at the Taum archaeota, and this is a taxonomic tree of the MAGs we reconstructed. What's highlighted in green color are the MAGs we reconstructed from the microbiome data, and all the other genomes, which are in gray here, are known isolates from the databases, right? And then you can compare these to the known isolates, and what you see is that these MAGs represent at least 3 different genera. So these are new genomes you can look at now. You can look at the function of these genomes and so on. And this is just showing a different example. This is a Nitrosphaera MAG compared to a reference genome. The reference genome is also, this is a type strain. And then you can start comparing these newly constructed genomes to the type strains. Right? So the type strain is in the middle here. Each ring is basically a different genome, and you can start looking at the genes, you can start looking at function and so on, and the difference of the different species. And then, of course, you can also map this to metabolic pathways. So what we do, we take the genes, we compare the genes to the KEK database, and then you can map it onto the pathways. So there's a pathway on the right, which I can't really see, doesn't really matter. So you can map it to whatever pathway you're interested in and see if this pathway is actually represented in the genome. And of course, you can compare data sets also. You can compare conditions based on these mappings. So you can look up at the abundance, for instance, the abundance of a particular genome, and then based on the abundance, you can look at the abundance of the function, right? Because if you have more of these genomes available in the microbiome, that might be a sign for a specific function which is important for this particular environment. So you can do a lot of things with the data, and you can spend a lot of time in looking at these genomes. And again, so we will make this available, the whole pipeline will be made available, and then you can actually start analyzing the data yourself if you have the compute resources available. Okay, so the next example is about plasmids, and I will hand over to Sören.
Okay, thank you very much. Yeah, so as Alex was saying, one of our main focus was to try to develop tools so that we can gain more information out of already existing metagenomic data. And the very big step forward is, of course, to actually assemble genomes so we actually can see which genes are actually in what organism. But we can actually take it beyond that. So when we start assembling, we can try to look at the close genomic organization. And in many cases, and what I just want to give you an example of here, that actually making a lot of difference. So, as you may know, bacteria can also have plasmids, independent replicating genetic element within the bacteria, and we know them for giving adaptive traits like antibiotic resistance and virulence, but they may carry many other traits. Those can be very difficult to identify in metagenomic data where you just chop up everything, but with a pipeline where we're doing all kind of tricks to assemble the contigs into full replicants, we can also identify plasmid. And we have developed and worked on having a pipeline available for that also now, so we have that on GitHub so everyone can look at it. So evidently, as I said, why antibiotic resistance is coming into mind. There's a lot of genes which may, if you just blast it, indicate that this is an antibiotic resistant gene, but it may not be. If that gene is sitting in the chromosome, it's actually just a household gene. That bacteria is not antibiotic resistant. Whereas if it's actually sitting on a plasmid in high copy number, it is an antibiotic resistant. So for One Health perspective, it's important to try to identify these. So I'll give you two examples. One where we were running our pipeline on some metagenomic data we had from the urban water systems, from wastewater sewers and wastewater treatment plants. So here we had 78 metagenomes we analyzed, and we characterized plasmids. So if you're looking at that Venn diagram, the green one is all known plasmids ever sequenced in the world until we did our study, and the red one is just that one study. So it's like almost a 30% increase of the amount of plasmids we know just by looking in one study. You will see there's a small overlap, but actually the vast majority of the plasmid we found were new one, never seen before. So there is a huge diversity out there. And this is another data set we have been looking at where it's 400 samples coming from the infant gut, where we have tried to assemble plasmids in these. And what the graph here will show you is that if we are looking for how many taxa we will be able to identify different bacteria, it's around 400 in that scale in the children that we are finding. But if we are looking at the plasmid pool, it's several thousand. And that means that we have here an indicator with a lot of more resolution for seeing differences in the children. So we were encouraged by this to look more into the data set and see does this actually correlate with something. So we actually were trying to trawl the internet for a metagenomic data set from early life gut microbiomes and also some adults. So we are ending up with more than 9,000 metagenomes, and around 7,700 of these were for infant. Identified all the plasmids here, and what this figure is showing is just a different type of plasmids. And what we will see is that there were some of these plasmids that we seem to find in basically all samples. Independent of it's adult or babies, we find this plasmid, they have it. But there's also plasmids, like the upper one here, they are only present in breastfed infants. And some plasmids are only present in formula-fed children. Something that is difficult to see a difference in if you're just looking in the overall microbial community, but the plasmid seems to be an indicator of this. It may not necessarily need to be because the plasmid are giving specific traits which are important for breastfed children. It may just be that they are sitting in a strain which have traits which are important. So we are down in a really subspecies strain resolution that you can get out of. You could, of course, get that by just sequencing maybe 100 times deeper. What we show is you don't need to sequence more. You just need to have the right indicator for this. So when we are looking at it, we also see that there's a lot of functional genes on these plasmids. Actually, Some of them we can put function to, but I think in future there's a huge task here to try to identify the function that more than 50% of the genes encoded on these plasmids, we have no guess of what they are. There are no similarity in the databases for any genes with these functions, but we have to remember that they are sitting on plasmids which are selected for their ability to give an advantage to the host. If these traits are not important, they would have been lost. So we have vital, important traits they're selected for all the time, and 50% of them, we have no idea what they do. So an interesting new window we are opening up here, and that was just one example of how more sophisticated analysis of the data we already have can bring us an important step forward. That was all we had from Work Package 1.